Hello, everybody, and welcome to a new session of the HPP Innovation Week, the benchmark event on high-pressure processing technology. This is Oscar Garcia, marketing specialist at Hyperbaric. And first of all, let me remind all the attendees that they have an open chat where we will be trying to answer all the questions that we receive. So during this session, we are going to discuss about the effect of HPP to control bacterial spores. And for this, we have here two great speakers. They are my colleague Mario Gonzalez. Hello, Mario.
Hi, Oscar.
And we also have Chris Duna. Hello, Chris.
Hi,
Great to
nice
have
to
you
see
here.
you.
Yeah, great to have you here. All right, so now let's start with you, Mario. So Mario is a doctor of food technologies working as food applications and processing specialist at Hyperbaric since almost 7 years. He obtained his PhD with his research activities focused on the validation of HPP as a safe approach for preservation of low acid beverages such as coconut water. Now he will explain it better. And at Hyperbaric, he combines his research activities with the provision of technical support to users of HPP technology. So Mario, all yours. You can start presenting your presentation.
Yeah, thank you, Oscar. You can see my screen, right?
Yeah, it's perfect. You can continue with it.
Okay, thanks. So yeah, as Oscar said in the introduction, I will just briefly explain my work. And this session is entitled Behavior of Clostridium botulinum spores in coconut water. Yeah, to have a clear idea of this, I will briefly explain coconut water. So from a commercial point of view, this is a huge beverage because it accounts for 96% of the total share of plant-based waters. And from a quality perspective, tender coconuts are preferred for coconut water extraction. The water content is higher and they also have a more aromatic flavor profile. Here you can see how a tender coconut looks like. Coconut water has some interesting properties. It is source of vitamins. You can see here ascorbic acid and many B group vitamins also. It is also source of bioactive compounds such as polyphenols, caffeic acid, procyanidins. And there are some research papers supporting some functional properties such as antimicrobial activity or even anti-proliferative activity against cancer cells. So according to FDA, coconut water is considered in the United States a fruit juice. Pure tender coconut water is a low acid juice because pH is normally above 4.6. And the juice hazard regulation requires process validation to ensure that control measures achieve a 5-log reduction of the pertinent microorganisms. Yeah, since coconut water is a low acid juice, Clostridium botulinum spores are considered the pertinent microorganism that the process validation should target. Now we can go a bit more into Clostridium botulinum because it is a spore former. Spores are highly resistant and dormant structures created by some bacteria in response to adverse conditions. They are very resistant to environmental conditions, including pressure. You can see that pressure levels above 10,000 bars are not enough to inactivate spores. And commercial HPP units, for instance, the highest pressure they can reach is 6,000 bars. And yeah, it also represents a food safety concern. If conditions are favorable, Clostridium botulinum spores can germinate, grow, and produce the botulinum neurotoxin. So, spores are the normal form of the bacterium in the environment, and they are not hazardous while dormant. Nonetheless, if conditions become favorable and in the presence of some key nutrients, spores may germinate. If conditions remain favorable, germinated spores become live cells and they start to replicate. And things become here dangerous because, yeah, some reports say that when the exponential growth phase is reached, the botulinum neurotoxin is produced. But some research is needed to fully understand if this is the exact moment when the toxin is produced or not. And again, if conditions become less favorable, those live cells become spores again. So this is how the bacteria disseminates in the environment. From a food safety perspective, we can classify Clostridium botulinum in two types, non-proteolytic and proteolytic spores. Each type differs in growth conditions. Since high-pressure processing does not inactivate bacterial spores, food safety authorities normally consider growth boundaries as a control in food. You can see here highlighted in red that the non-proteolytic type grows even under refrigeration, and the proteolytic type grows at pH 4.6. It has the potential to grow at this pH level. So these are the boundaries normally considered for food safety. And yeah, I mentioned before that coconut water is a low acid use, so FDA requires additional control if high pressure processing is used as a control method. Okay. And here comes our research question. Is really Clostridium botulinum the pertinent pathogen in coconut water that process validation should target? There is a lack of growth, no growth models in coconut water. There is also lack of epidemiology evidence, and other intrinsic and extrinsic parameters in addition to pH and temperature play a role in the growth dynamics of Clostridium botulinum spores. So to give an answer, or to try to give an answer to this question, we proposed the following experimental work. We selected a wide range of coconut waters, up to 8 from different locations and varieties. We sterilized all coconut waters using a cold gamma irradiation. Background microbiota could play a role in the growth of Clostridium spores. So to remove this variable, we sterilized the coconut water. We also adjusted the beverage to different pH values just to create more favorable conditions. And we also played with these other parameters. Redox potential was adjusted to create very low values that really support Clostridium growth. We tried to supplement coconut water with a combination of vitamins, mineral, amino acids, or PYGS broth. This is a laboratory media which is very rich in nutrients. We prepared samples in different volumes depending on the experiments, and we worked with two separate cocktails of Clostridium botulinum spores, one with 9 proteolytic strains and the other one with 9 non-proteolytic strains. And yeah, those strains are very well documented in the literature. And they tried to cover a very big range of subtypes of Clostridium. Incubation was made under anoxic conditions at 30 degrees up to 50 days. So these conditions really support Clostridium growth. And yeah, we checked this growth based on gas formation, the detection of the toxin genes, and also by ELISA to detect any toxin if it was produced in coconut water. So yeah, I have some tables here with the 8 types of coconut water, and you can see results for the natural pH of coconut water, a slightly higher pH between 5.5 and 6, and a combination, a mixture of coconut water and PYGS broth. We did that as a positive control. Actually, you can see that we found the toxin genes in all blends of PYGS broth and coconut water. Nonetheless, we were not able to detect growth based on gas formation or toxin genes in the natural beverage or even in the pH adjusted. We only found toxin genes in one type of coconut water adjusted to a higher value. We call this type T2. Okay. So for future experiments, we focused in this particular type of coconut water, and this time we played with a bigger pH range. Okay, you can see that we also have the natural pH of coconut water, and we adjusted things up to pH 6.4. We tried to adjust the combination of coconut water and PYGS broth to the same values, and we also worked with the broth, with PYGS broth. Incubation was made for 50 days at 30 degrees, and here you see the results. We were not able to detect toxin genes and gas formation at the natural pH of coconut water, and this time we failed to detect also the toxin genes at pH 5.5. However, adjusting the coconut water to higher values gave us positive results for the first time. Okay, we could detect the toxin genes for both Clostridium types, and also we saw some gas formation. And again, the combination of PYGS broth with coconut water worked as a positive control. At all pH values, we could see growth of the spores. So these results may suggest that coconut water may be a source of anticlostridial compounds, or that acidity of the beverage and nutrient content determine spore growth. To validate this hypothesis, we still work with T2 coconut water, and this time we adjusted the juice to the highest pH value at which growth was not previously detected. And here you can see that for proteolytic strains it was 5.5, and for non-proteolytic it was 6. This time we supplemented coconut water with decreasing concentrations of PYGS broth, and we checked the time that it took for Clostridium to grow based on turbidity. So turbidity was detected in coconut water when very low concentrations of PYGS broth were used to supplement the beverage. This suggests that coconut water is not an antimicrobial source for Clostridium, but it suggests that some key nutrients that Clostridium needs to grow are missing actually in coconut water. So to have more precise results, this time we tried to include the detection of the botulinum neurotoxin with the ELISA test. We selected 5 representative types of coconut water, and we adjusted this natural coconut water to different values. Non-proteolytic Clostridium botulinum spores failed to grow at the natural pH, but even when we adjusted the juice to high values. We only saw some gas formation, but as you can see, we never detected the toxin genes or the botulinum neurotoxin. So this gas formation is maybe some abiotic reaction that we found in the media. Actually, the Sri Lankan type of coconut water did not support growth at any pH value based on any of these indicators. None of the coconut waters studied supported growth of proteolytic Clostridium botulinum at its natural pH. Only the type T2 of coconut water supported this growth based on toxin detection with ELISA. Nonetheless, you can see that the amount of toxin that was formed is very tiny. It was around 10 mouse lethal doses. Yeah, there was an outbreak linked to carrot use, and you can see that the amount of toxin was almost 100,000 times higher. And you can see that the human lethal doses is 3,000 mouse lethal doses. So yeah, as a consumer would need to drink up to 300 milliliters of coconut water to have the disease. This is just an assumption based on our results. And again, yeah, adjusting coconut water to higher pH values gave positive results for other varieties. So in conclusion, under highly favorable growth conditions, growth and toxin formation by proteolytic Clostridium botulinum was only reported in one type of coconut water, but none of the coconut water studied supported growth by non-proteolytic spores. This poor growth and toxin formation in natural coconut water can be attributed to a low pH and nutrient limitation in coconut water. So this combination of factors uh, create a really adverse environment for Clostridium to grow. The highly favorable growth conditions that we used in this study do not resemble at all commercial storage conditions for HPP coconut water. Remember that HPP beverages are always stored in the fridge, okay? And this study was conducted at 30 degrees. We also sterilized coconut water. This is not a normal scenario you would expect after HPP in coconut water. Therefore, we can somehow conclude that Clostridium botulinum may not be considered the pertinent microorganisms in tender coconut water for process validation. Uh, there have been several outbreaks associated to Salmonella, for instance. So maybe it's more interesting to validate the process to control Salmonella rather than Clostridium botulinum. Now that we have seen that spores really struggle to grow in the beverage. Well, I hope you enjoyed my presentation. This was all from my side. And now I will give the word to Chris Duna. Thank you.
Thank you so much, Mario, for the nice presentation. Yeah, sharing your research here with Clostridium, specifically in coconut water. As you said, now it's time for Chris. So yes, Christopher John Duna is a senior research chemist for the U.S. Army and Native Soldier R&D Center, and which is a very unique type of research institution covering basic and applied research development and testing activities for a wide range of mission-focused applications. Kiriduna is a doctor as well and has a broad experience working with non-thermal technologies on spores. And among his studies, he has analyzed precisely the effect of HB on spores. So, Chris, I've seen you are sharing already the presentation. Yes, the floor is yours.
Thank you very much, Oscar, for the kind introduction. And thank you, Mario, as well, for the introduction and the great presentation. I really enjoyed that. I'm going to have some questions for you in a little while after this one and look forward to discussing with you high pressure and with you. So thank you again for the invitation and great to be here. So my name is Chris Duna. I'm going to talk about the quasi-chemical modeling of bacterial spore germination by high pressure. I am here representing in a personal capacity, which is really great because I find this work personally very interesting. It has a long legacy. I worked with Florence Fihiri at Natick, great research microbiologist, prepares some of the purest spore crops. She actually worked for Dr. Hill Levinson, a Natick Hall of Famer, who's one of pioneer spore researcher from decades ago. So it's a real privilege to work. It was actually somebody that I think Peter Sello looks up to. We had them both at a review one time at Natick. So it's a real privilege to be here. We actually refer to Hill's papers from the 60s in some of our current work. And again, thank you so much for the invitation. So I work in a lot of different research areas within Natick, everything. Oscar's mentioned the non-thermal technologies and food preservation methods, but also in textiles, clothing, shelters and tents, preventing mold, on parachutes and sleeves, and as well as chemical and biological protection. And we work with a number of universities in Massachusetts, in I guess it's Boston, Worcester area, MIT, UMass Lowell, Worcester Polytech, and Brandeis. So very fortunate to be where we're located and to have such great collaborators. Just a quick side note here is I developed a chlorine dioxide producing product that we patented and licensed and sold commercially, and it was used during the Ebola crisis. Now, it's probably about 6 or 7 years ago. It can also be used to sterilize lab equipment, kill spores, and that can be like a biohood in that center figure, or a spore bioaerosol chamber on the right that we use for chem bioprotection. So I just want to briefly mention that, but really I want to talk about high pressure processing. We have 2 units at Natick that I've used. One's a 2-liter unit on the left-hand side. It doesn't quite go up as high in pressure and temperatures. It works great though, great unit. And we also have a small kind of kinetics unit on the right-hand side there, a 20 milliliter unit, much smaller volume, much more efficient heat exchange with the environment, but also more efficient come-up time to high pressures. And we're going to talk about the impact of that on some of our spore results a little bit later. So high pressure processing, it's a field we've worked in, great field to work in. I actually started out working in high pressure as I was doing some inorganic chemistry in the chemistry department. We were looking at metal complexes and the effect of high pressure on the kinetics of metal complexes. And lo and behold, I came to Natick, and it was a high pressure machine, and then it turned into microbiology and food processing. Preserves food. And I think Mario was saying it's got a fish-like character of the coconut shows, higher nutrient retention. The challenge of eliminating, it can eliminate vegetative pathogens. Spores are a little bit more of a challenge. And that's really what I'm going to talk about here. We've also used it in addition to foodborne spores, but on Bacillus anthracis stern. That's a surrogate for anthrax, and that was in connection with chem bioprotection. So another thing I wanted to mention, just from my first book on high pressure in 2005, we had a comparison, a chapter done by Kraft, of salmon fillets. They were done by conventional retorting, and they came out they were safe, but they were kind of dried and discolored, whereas the salmon fillet cooked at high pressure also killed the spores, retains its pink color, higher sensory attributes, juicy and tastier. So that's some of the benefits of using high pressure. And that's just one of the earliest and great examples from Kraft. For our study, we inoculated whey protein solutions, where it could be vegetative pathogens or spores, sealed them in a bag, put them in a high pressure machine, and then plated them out afterwards to enumerate survivors. And we're going to look at a range of different organisms, both pathogens, E. coli and Listeria, through spores, Bacillus subtilis, amyloliquefacients, and even Clostridium difficile. One of the kind of unique challenges is that to do the kinetics, right, during the come-up time, both the pressure and the temperature are changing. They're not constant, right? It's going from ambient pressure to tens of kpsi. So what we did is in order to get normalize our kinetics, we took enumerated the survivors just at the come-up time before the hold time of the high pressure. And we noticed at the lower temperatures like 30 and lower pressures 30 kpsi, we didn't see much change. We go up to the higher temperatures, 50 degrees and higher pressures. This is for E. coli and whey protein. 50 degrees Celsius, 60 kpsi, we saw it was only about a 34% survival. In other words, you can get substantial inactivation just during the come-up time. But it does depend on the organism, food substrate, pressure conditions, and so on, and the equipment you're using. If we took then the kinetics just during the hold time, what we see is that at lower pressures and temperatures, you get a lag time and a slow inactivation rate. You increase the pressure, you get faster, more linear inactivation rates. Makes sense. Same thing with temperature, a lag time and slow connects at low temperature, increase the temperature, shorten the lag, increase the inactivation rate to linear inactivations and faster rate at the highest temperature. I think it all kind of makes sense. The model we use is called the quasi-chemical model. This model, it's actually over parameterized for those kind of inactivation kinetics, but it's a growth death model that we've used and based on differential equations. We've used it for hurdle technology and shelf-stable foods. We've also used it for the high pressure. It's great because it can incorporate some of the nonlinearities that are observed in the kinetics. Another organism we looked at was a barrel-resistant strain of Listeria monocytogenes. And again, we approach it the same way. And we see something also very similar, that at the low pressures and low temperatures, during the come-up time for the Listeria and whey protein, there's almost no inactivation. And that's true at the whole low temperature regime. We go up to the higher temperatures, 50 degrees, and go through the pressure regime. We see, maybe not so much inactivation at 30 kpsi, but only 1.1% survivors at 60 kpsi. Now, these are not your typical processing conditions, I acknowledge, but it did give us a chance to study the organisms and the behavior and learn how to handle it. It becomes important for how we handle spores later. Again, we see the same kinds of things, a slower inactivation rate at lower conditions, increased conditions, faster inactivation rate. It's just that in some conditions at the bottom of the slide, we saw tailing, long persisters that weren't killed by the process. And these lasted for tens of minutes. I think we're out to about 60 minutes there. The model we use for that, if we look in the top left, is a 6-parameter model. It's a growth, death, tailing model, what we call the enhanced quasi-chemical kinetics model. And again, I mentioned it's over parameterized. So if we look at the right-hand side, that can be made more parsimonious into just 4 parameters, and that will be sufficient for us to handle the inactivation kinetics showing tailing. They're basically the same model, but it just makes it numerically easier to handle by scaling it down. We also innovated some other stuff, a transition state theory from chemical kinetics, looking at the tailing data, using what we call a processing time parameter, the time to kill 6 logs of listeria in the food with high pressure. And these are just kind of conventional transition state theories. It was a very clever innovation by my colleague, Professor Ken Custon from Brandeis, who's a chemical kinetics expert. Just shows you the diversity of fields being used in this work. Now, spores are the real challenge, and that's what we're going to get to here. And Mario gave a great lead on, well, we have spores, you have a germination, sporulation, but things are not just single-step processes. They're complex, multi-step processes, and a great field for anyone who loves to work in it. Or anyone who works in it, it turns out they love to work in it, I should say. One of the things is activation, and that's where the spores are potentiated to germinate. So we'll see some examples of that. Germination, as Mario mentioned, they go from dormant to metabolic activity, but that's a multi-stage process, commitment, stage 1, stage 2, all these different parts of the stage going on. And then there's inactivation. I'm going to come back to inactivation. I want to point out back with respect to germination, at the lower pressure, there's lower pressure conditions, you can induce germinations by stimulating germinant nutrient receptors. You go to that's at about 150 megapascals. At about 400 to 500 megapascals, you stimulate germination by stimulating the Spore protein channels. So there's different routes to germination, and a lot of different things can go on. So let's take a look at what we saw. With respect to activation, if we put dormant spores in the whey protein and then put it in a heat bath, we look at the top left, just at some high temperature conditions for a series of minutes, we can see an increase in the population. In other words, some of the super dormant population is activated and then can germinate when plated out. We see that with temperature, that's well known. On the bottom right, we saw if we do it at high pressure, high temperature conditions, we also get activation just by in the come-up time of the equipment. They're exposed to a high temperature and high pressure profile. And we see here in all those turquoise colored bars are the increase in population occurring just from the come-up time of the sample being in the high pressure unit. This is at before the hold time of the high pressure and not even in the kinetics, but it means your actual counts are changing. If we go to germination, now we're going to see here a little bit different change, because this is we're looking at Bacillus subtilis spores. They can germinate. They go from phase bright to phase dark. Again, if we go with the 150 megapascal conditions, stimulate nutrient germinant receptors. 550 megapascals stimulate the Spore proteins. In both cases, they germinate. In both cases, the model, which is the straight lines here, are able to fit the data. The germination model that we came up with is a very scaled down version of the multiple steps in germination. The high pressure stimulates the spores, and a stimulated spore can either germinate or remain super dormant. There's not a lot more to elaborate on that, because I think we need to do more mechanistic work, but this model works well. Well, what do I mean? Here it is. We look at Bacillus subtilis in high pressure, 150 megapascals, a series of different mutants of Bacillus subtilis. And they came out of Pete Sutlow's work at Connecticut. And the mutations influence proteins and how well the spores germinate. But we do see they germinate, albeit some faster and some slower, based on the mutation or whether it's a wild type. But we do see that the curved line fits the data well in all the cases. We move up to the 550 megapascals. We see that a lot of those mutations don't influence germination here. So those are probably proteins that influence nutrient germinant receptor germination. What we do see, however, is that the data shows germination and the model fits the data really well. If we look at some of the slides in the bottom right, we see that the mutation does retard germination slightly. Model still fits it well. That's what we were looking for. One of the big changes is we looked at Bacillus liquefaciens. Again, we see germination occurring very fast with high pressure, either 150 or 550. But Clostridium difficile spores, They don't go from phase bright to phase dark. It's different physiology. We had to do a DPA release measurement, collected that data, and the model fit the data well, all the same. This is, I would say, sort of related. It's in the Clostridia field, like Mario was looking at. And also, it just shows you some of the diversity within spores that always pose some different challenges, not a one-size-fits-all approach. If we look at that same model I was talking about earlier, growth, death, tailing, kind of enhanced quasi-chemical model, but we look at the inactivation now during the whole time of a high pressure processing, there's a couple of things that are really important. Firstly, the prevalence of tailing. In other words, it's a super dormant population that is not inactivated over the course of time. Now, the way you would have to get rid of that is go to higher pressure and temperature conditions. But for the sake of characterizing the behavior of the spores, we see that the tailing behavior lasts out for up to 200 minutes, I think, in some cases. Not a practical processing time, but a learning tool for how to process better. I think the top two graphs show one of the best comparisons that I like. We used our 2-liter unit, that's on the left-hand side of the EPSI unit. We got about a 2.5 log reduction at high pressure conditions. All identical conditions repeated at our 20 milliliter volume, we got a greater than 4 log reduction, exact same conditions. And the implication to me is that when you take measurements on small machines, I understand that it's easier and better to handle, but they may not extend to larger machines. And I think it just means you need more validation at large machine and processes, as well as in laboratory for lab setting equipment. One of the things, so I just want to close out mentioning some SEM images of the Bacillus amyloliquefaciens spores. Just like Mario described, you see in the kind of ovoid structure, plump and intact. You see everything there. These are untreated spores. You put them in an autoclave and they disintegrate, all structure gone. They're inactivated, kind of almost no question. You really believe that. You put them in a high pressure machine, again, high pressure, high temperature. The structure of the spores is damaged. They're all withered, and those are also inactivated. I'm not going to show it, but I have chlorine dioxide treated spores. They look untouched, but they are quite killed by the chlorine dioxide. Again, just a total different mechanism of killing the spores. So the conclusions really that I want to just kind of sum up is well, there's different pathogens and obvious different model forms and different things that will happen. We are able to show that, the quasi-chemical model works for E. coli inactivation. We can modify the QC model to do Listeria monocytogenes that shows tailing. We do see, though, we can also use that same enhanced model for spores, where we see activation, germination, and inactivation, and sometimes all just during the come-up time, depending on the conditions, just during the come-up time. So we're touting our 3-parameter germination model, showed it is very flexible for a range of B. subtilis mutants with different proteins and so on, different HPP conditions. But it needs further work to characterize those mechanisms and integrate them in with the models. So it's not just a skeletal model, but can be more detailed. But these models are useful for validation studies and finding other surrogates and modeling the inactivation kinetics with HPP and other non-thermal technologies. I want to close. This is a colleague, Mrs. Florence Viheri, who did, I mentioned she prepared some of the greatest spore crops, cleanest and so on. Great work in Natick, did a whole bunch of contribution to this work, was actually a student intern with Hill Levinson not that long ago. So there's a huge legacy of some really terrific spore people, great collaborations with Peter Sutlow and so on. Really fortunate to be part of it. Some of the books of the past. I just want to say I really appreciate the work that Mario is doing with the Clostridia. They're tough to work with, but on the coconut water, just showing how HPP can be implemented and some of the challenges and difficulty of dealing with spores and real applied work. With that, I want to say thank you very much and happy to take any questions and discuss with you anything you like.
Well, thank you, Chris, for your presentation. It's great to hear to such a broad, a person with such a broad experience with this topic. And now, I guess you want to discuss a little bit about precisely this, your different research and studies. So just feel free to have a, informal conversation, less discussion among professionals on this topic. So any questions you have, Chris, for Mario, and Mario for Chris, just feel free to share it.
I'd like to start if that's okay, because I do look at the germination. There's so many different steps in the sequence. I mentioned going back all the way to the Hill Levinson work, but you point out, Mario, a bunch of different strains, and one of the questions I was wondered is, where in the growth cycle do these strains produce the toxin? And I've seen papers where it can occur in different parts of the, even in the early phases during the germination phase. What have you found about that, and how does that really influence your approach?
Yeah, this is a very good question, and it's not easy to answer. Yeah, to be honest, I didn't focus in this particular aspect. Each strain should be characterized for this, I think, um, and this was not my work. I also think it depends on the metrics you are working with. Maybe the strain produces the toxin uh, at one point in the life cycle in one media, but if you change the media, the toxin is produced a different time. I don't know, but yeah, I think more research is needed to fully understand this variability, and yeah, to design process validation. I think it's important to consider this.
Okay, I mean, but so your real concern though is not what happens when it germinates, is preventing it from be there to germinate in the first place, right? Is that your strategy more or less?
Yeah, that is our approach. That's what we try to demonstrate somehow. So it is important to understand these aspects to continue this approach, I think.
I was also kind of interested. At first, it struck me, this was when I found out some years ago, that Spore was a pathogen of concern for coconut water. I was actually wondering if there were others. Um, are there others? And is it just the fact that the water is taken like straight from the coconut and not treated further, or are there other pathogens of concern?
Yeah, coconut water production is very simple. You just have to crack the coconut, collect water, fill bottles, and process it. So regarding other pathogens, yes, I mentioned that there are some outbreaks with Salmonella or even Vibrio. Okay, but I think this is more related to fresh and non-processed coconut water that was temperature abused, and those pathogens have the chance to grow to high levels and cause the disease. Um, but with Clostridium botulinum, yeah, it really needs some time to adapt, to grow, to germinate. So I was not able to find any cases related to this. Mhm.
Yeah, you say that it's easy to just crack the coconut and get the water, but I think I saw in Brazil they were slamming these on the sidewalk to get them, and I don't think
Yeah,
it's that easy.
no, no, of course it's not. So it's also important to work on these clean conditions for the food production. Um, yeah, to collect coconut in a clean condition, to fill as soon as possible the bottles, and to process them also very quick. Yeah, those things, if are well implemented, they help to reduce the risk, of course. Mhm.
And the final kind of thing is, it seems to me, and this is a guess, it's a wild guess, is that generally to kill spores or to substantial levels in particular, you need a lot of pressure and temperature. But I don't. I think that would really compromise. Perhaps it could compromise the quality of the coconut water. Is that what you think too, or because it seems? I guess your comments on that in general.
Wow, yeah, this is something we have not tested because combining high pressure and high temperature is very challenging, as you know.
Yeah,
Uh, so I think this is still far from commercial implementation, although we are working on some solutions to make this process feasible. Okay, we have discussed this in other sessions, I think. But I agree with you that combining pressure and high temperature will change the properties of coconut water. It's a very delicate juice, so for sure you will change color, and most likely you will change flavor. So the good thing of high pressure processing is that you can maintain these color and flavor properties as the fresh beverage.
got it. Yeah, that sounds good. It may not need to be as rigorous or vigorous conditions, and you still
Yeah.
may be able to achieve the
Mhm.
the safety that you're looking for.
No, you're right.
One of the things, and and it looks like, I guess the other kind of question was you only focus on the CBOT. I get it. But have you looked at bacillus type at all, or is that just simply not an area of concern?
Of course, it is a concern. I haven't looked into it, but it is also a concern. I guess it also can grow in the beverage if the right conditions are given. So, as a future work, I think this is something we should consider. But yeah, it seems that food safety authorities are more concerned with the Clostridium botulinum because it seems that yeah, the disease from Bacillus cereus, for instance, is not so serious. So yeah, the main focus is Clostridium botulinum. But yeah, I think all spore formers should be considered for a risk assessment. Mhm.
And I guess the final kind of question um, is, I'm sorry if I asked too many questions. I thought, if you don't mind,
It's fine. For the
but
audience, this is great.
what what's how much has selection has to go into the type of packaging or bottling that the coconut water will go into, and does it matter what the type is, and that kind of influence. Does it matter to the quality of the product, to the safety of the product, even to the processing if we're going to be high pressure?
Yeah, for high pressure processing, you will need waterproof material, flexible and elastic. So the most common one is plastic, of course. And this could play a key role because you have different types of plastic. Oxygen transmission rates are different for each plastic. And as an anaerobe, Clostridium will behave different if you use one type of or the other type of plastic. Again, this is something we can look into in future work. So far, we have worked with PET. PET is the most common plastic material for HVP uses, and it is a very good barrier toward oxygen. So, yeah, the transmission would be very low. Um, but if we play with other plastics, maybe we can include this oxygen transmission as a new variable to consider. So yeah, your questions are very nice because they show that there is still a lot of work pending. But yeah, it's not a simple issue. Mhm.
Well, yeah, okay. So is it approved anywhere? Then, coconut water with high pressure treatment, and
Yeah, um, you can find it, for instance, in Europe. In many European countries, you can find HVP coconut water. Same goes for Asia. We have some customers commercializing um, HVP coconut water. Of course, the risk is there because pH is high, but I guess it depends how you assess this risk. The product is always in the fridge. pH is close to 4.6. So I guess some food safety authorities consider that somehow the risk is controlled, and this is why they allow the commercialization of
mhm,
HVP coconut water. Yeah.
mhm. And how? I mean, you did mention, to jump back a little bit, about the plastics and the anoxic conditions. How sensitive to oxygen? Just how sensitive to oxygen is the coconut water?
Wow. This is a nice question because, for instance, when you heat pasteurize a product or coconut water, you are removing a lot of the oxygen it has dissolved. Okay, but with HVP you do not, uh, because you don't rise temperature. You have actually high dissolved oxygen content. Um, this can play a role in the growth dynamics of Clostridium spores. And yeah, it also is funny in the case of coconut water because it oxidizes. The oxidation of coconut water makes it pink. Um, it's a funny discoloration, but consumers associate this with a fresh product, so it can be used as a marketing strategy. But again, uh, this is one thing to consider in
Uh,
process validation.
Oh, that's great. That's funny. I'm not so
See,
sure. I think
I don't have many specific answers because there are so many things to look into, and yeah.
of course, those are great. And the thing that the other thing, and this is on a little bit side note, is you did talk about some of the equipment. I do remember the first time I saw the horizontal units from Hyperbaric. I just thought they were absolutely amazing. But the question I have is, and you referenced this, is really going to high pressure, high temperature conditions simultaneously, right? It wears out valves sooner, it doesn't have the same throughput, and so on. But
Mhm.
what is like the possibility for that? Is that something that's always kind of capable but not in demand? Will it increase when there's more demand? Can it go to higher pressures and temperatures now? What does the situation look like for that kind of high pressure, high temperature processing?
Yeah, good news is that you can actually do this. Commercial machines cannot handle high pressures and high temperatures uh, because now temperature becomes a critical control point. So in a high pressure vessel, you have gradients of heat, um, and this is something very hard to control. But in collaboration with the CSIRO in Australia, they have developed a plastic canister, okay, that creates some insulation from the vessel. So the idea that it can be done nowadays, because it is already validated, is that you can preheat this plastic canister. You fill the plastic canister with hot water and preheated products. Then this canister is closed and placed in a conventional HPP machine, and you can actually pressurize the whole canister. So this is a nice way to apply HPP, and because of the isolation of the canister, we are also controlling temperature inside. And yeah, we have validated this, and there is no gradient at all inside the canister. And this is something that can be done in any commercial HPP machine.
I'm sure the Australians appreciate the plug, so good job there. The other, I'm just teasing. The question, this is my last question, but you were talking earlier about doing everything in individual bottles, right? Is that still just the model for how to process this product, or do they ever do it like a juice in bulk and then bottle it after the processing?
Yeah, well, good question actually. Now, yeah, the only way to process coconut water nowadays is in bottles. That that is what companies still do. They
Mhm.
fill bottles, they apply HVP to the bottle. But of course, we have this in bulk machine that can process liquids in bulk. And yeah, I guess coconut water can benefit from the process, but again, pH is high, so it is very important how you handle the
Yeah,
coconut water after processing. You need aseptic lines, I think,
yeah.
to fill bottles in aseptic conditions. And yeah, this is very challenging and expensive. But of course, for anyone with big productions and with a sterile filling line, they can go for it. But no one is doing this so far.
Yeah, I did. That's sort of what kind of I thought the answer most likely. I
Hmm.
wanted to just mention that. I'm really thank you so much. I thank you for so generously answering and discussing with me all this work and all your questions. I don't have any more. Unless there's particular questions for me, I think Oscar, not to cut anything short, I think it's okay if we wrap up. But thank you for being so available and helpful, Mario, for
No,
answering all the questions and discussing. I really appreciate and enjoyed it. I thought it was great
it's
work, and it's always great to interact with you guys.
no,
Okay.
for me it's a honor to have you here saying these nice things, because you have worked very hard in with HVP and sports, and also you have worked with many people that yeah, you mentioned some of them, and for me those are like the greatest researchers on sports and HVP, and for me it is a honor to have you here and to say these nice things about our work. So
Uh,
congratulations also for your work.
well, thank you. The honor's mine, I can assure you, but thank you so much. I'm very
Mhm.
flattered. I do want to say thank you. And I guess if there's any other questions, or if you get audience questions, feel free to send me an email. And I want to, I'll look forward to talking to you guys later. in the week. But for now, I just want to say thank you again and wish you all the best and time to sign off.
Okay, thank you,
Okay.
thank you so much for both of you and the spontaneous discussion you have at the end. I think that these are the things that really add value to these kind of sessions and for the audience. So thank you so much for being with us today.
Okay. Thank you. Have a great day. We'll
Thank
talk to you
you.
soon. Take
Same to
care.
you.
Have a good
Bye.
day. Bye bye.